Hello, I'm Marysia. I'm one of the non-invasive cardiologists and a BIRCWH scholar here at Mayo Clinic, and I'm happy to be your moderator today for the BIRCWH Leadership Webinar Series. The title of our talk today is Reveal, Don't Conceal, Transforming Data Visualization and Statistics Education to Improve Transparency. And our speakers today are both former BIRCWH scholars, Drs. Stacey Winham and Stacey Winham and Tracey Weissgerber. Dr. Stacey Winham is an associate professor of biostatistics in the Department of Health Sciences Research. She was a BIRCWH scholar from 2013 to 2015 and is currently the recipient of the Walter and Evelyn Simmers Career Development Award for studying ovarian cancer. She is also an active educator, the biostatistics curriculum director for the Mayo Clinic Graduate School, and has a specific research interest in sex-based differences in genetics, specifically in the X chromosome. Dr. Tracey Weissgerber is an assistant professor and researcher in the Division of Nephrology and Hypertension here at Mayo Clinic. She is also a group leader and meta-researcher at QUEST, which stands for Quality, Ethics, Open Science and Translation at that Charité University in Berlin and also the Berlin Institute of Health. She joined Mayo Clinic as a BIRCWH scholar in 2012, and as a vascular physiologist, her research interests specifically align with studying eclampsia and the development of cardiovascular disease later in life. She also conducts meta-research designed to improve the quality of data presentation, statistical analysis, and statistics education in basic science. Drs. Weissgerber and Dr. Winham's meta-research paper on bar graphs has been viewed over 300,000 times and really gives a very essential information of how to present data. Today, the focus will be discussing some of those tools and resources along with other tips that they have to share. And I would also like to point out to you in the chat box, there will be a Twitter link for those who use Twitter with additional resources that you may find useful. And I'll hand it over to them because this is going to be a great topic and I'm looking forward to hear what they have to say.
All right, thank you very much. So I'm just going to share some slides because it's hard to talk about visualization with no visuals. So obviously we need to have slides here. Okay. So thank you for that introduction. As former BIRCWH scholars, Stacey and I are really excited to talk to you today about this work that started actually as a collaboration when we were both in the BIRCWH program here at Mayo Clinic. So, okay. So before we get started, I think it's important thinking about, why is it important to care about visualization? Rigor and reproducibility involves a lot of different things, from our methods to our analysis to our reporting to whether or not we're sharing our data and how we're doing that. And visualization is just one piece of that. So why is it a piece that it's worth taking the time to care about and really think about critically and make changes in? And I would argue the reason visualization is really important is because data presentation is really the foundation of our collective scientific knowledge. So when we think about how we publish and how we share our research findings, we usually use figures to show the most important pieces of data in our paper to help our readers see and understand what it is that we found. So the figures are especially important because they show our key findings. And it's also important to remember that most of the time the raw data underlying those figures aren't available. So most people aren't uploading or sharing their data in an open data format, and even when data are available, they may not be deposited in a form that we can really actually reuse them or understand what it is that's been shared. And if you email authors to ask for their data, the likelihood of receiving a response declines precipitously with the time since publication. So until open practices become more widespread, everything that we know about the data presented in a paper is often limited to what the authors present in their figures and tables. And if we're systematically making poor choices about the types of figures and tables that we use, that affects the availability and our ability to understand those data, not just at the time the paper is published, but also for future generations. So today we're going to start off with a quick overview of some problems with our current practices, focusing on bar and line graphs. I'm then going to turn things over to Stacey, who will talk a little bit about problems with statistics education, why this happens, and what we can do about it. And in the last section, we'll focus on solutions. So I'll talk about some solutions for static graphs, as well as some free online tools we've created for making interactive graphs for your scientific publications. And I'll include a little bit of a discussion on some of those resources. So the work that Stacey and I do predominantly focuses on small data sets. So in the era of big data, why is it worth spending time thinking about these small sample size studies? And there are a few reasons. The first one is that small sample size studies are very common in basic biological sciences as well as in translational science. So we know that in physiology, about 75% of papers, the largest sample size for any figure or any group shown in a figure is 6 independent observations per less. And that was the smallest sample size. The largest sample size is going to be 15 independent observations per less or less. So understanding what's going on with these small sample size studies is really important. In addition, the small sample size that are used in preclinical research often influence decisions about what potential treatments advance to clinical trials and further research. And we know that clinical trials are very expensive to fund. They place a lot of burdens on our patients, as well as on research staff, and they drain resources from other areas. So it's really important that we have as much information as we can about the preclinical data so that we can make sure that we're advancing the studies or the agents that have the best odds of success. And finally, it's important to mention that when NIH is highlighting problems with reproducibility, they're particularly focused on the preclinical studies. Clinical trials have undergone a number of changes in recent decades to eliminate some of the reproducibility problems that are still common to this day in preclinical research. Effective figures in a scientific publication need to do 3 things. The first one is to immediately convey information about the design and statistical analysis. The second one is to illustrate important findings. And the third one is to allow the reader to critically evaluate the data. And it's this third point where the types of graphics that we use in the basic biomedical sciences and in many other disciplines often fall short. I think it's important to emphasize that my interest in data visualization comes from problems that I see in my own area of research, which is preeclampsia. So we know that preeclampsia is a very heterogeneous syndrome. All women seem to end up with a similar set of symptoms, but they get there in very different ways. Some women have maternal pathways, some women the pathways are more fetal, and in other women the pathophysiological processes are more related to the placenta. And any woman may have any combination of those different things. What that means for us as preeclampsia researchers is that for any process or biomarker we look at, we expect it to be very abnormal in some women with preeclampsia and perfectly normal in other women with preeclampsia. So the research in this area is really focused on identifying subgroups of patients with different types of pathophysiologies and disease. And a lot of data are presented in bar graphs. There are no subgroups in a bar graph. If you want to understand heterogeneity, you need to use more informative visualizations to present your data. A bar graph is what you use when you want to mask or hide heterogeneity. So as was mentioned in the introduction, we started off by publishing this meta-research paper in PLOS Biology in 2015. This was viewed more than 100,000 times in the first month of publication, and since it was published, it has contributed to policy changes in a number of journals. So that includes journals like PLOS Biology and eLife, as well as the Journal of Biological Chemistry, and other journals are now starting to implement similar policy changes as well. So for example, Nature Publishing Group recently came out with a policy change that covers all of the Nature series journals. The paper included a systematic review where we looked at what types of figures authors were using to present their data. So we looked at all the original research published over a 3-month period in the top 25% of physiology journals, which is about 700 papers. And we reviewed each of those papers to see what types of graphs they were using to present continuous data, because that's what transatlantic flights and jet lag are for. And when we did this, we found that almost all papers are using bar and line graphs to present their continuous data. So 86% of papers had a bar graph, 61% had a line graph. When it came to more informative figures like dots plots, box plots, and histograms, those were very rare. So only 13% of papers had a dot plot and 5 to 8% had a box plot or a histogram. We saw, as I mentioned before, that the sample sizes are very small, in most cases less than 10 independent observations per group. We saw that when investigators are using bar graph, they're typically using the error bars to show the standard error. So 78% of papers were using standard error bars versus only 16% showing the standard deviation. So most of the time our error bars are telling us about the precision in the mean rather than the variability in the data. And we also found that more than half of papers that were using non-parametric analyses presented data that they analyzed with non-parametric tests as mean and standard error or standard deviation. And this is a problem because non-parametric tests are usually what we use when we don't know the data distribution or we know that the data aren't normally distributed. So the standard error and standard deviation and the mean can all be very misleading in these circumstances. So 85% of papers contain a bar graph. Why is this important? Who cares? Well, the reason that we should care, or one of the reasons, is that many different distributions can lead to exactly the same bar or line graph. And the actual underlying data may give us a different impression from just seeing the summary statistics alone. So here we have an example where we have a bar graph on the left, and then we have 4 different data sets on the right that could lead to that same bar graph. When we look at the first data set, we see that the observations in the second group appear to be slightly higher than in the first group. So this might be something that we're interested in exploring further. When we look at the second data set, we see that those higher observations in the second group are mostly driven by a single outlier. So it's much less likely that we would be interested in pursuing this finding. In the third data set, there's some indication that our data might be bimodal. Now, the sample size is really small, so it's hard to tell whether that's actually true, and we want more data to determine this. But if the data are in fact bimodal, we would want to know, is there another factor that's explaining what's going on in this data set that we need to account for? So for example, do the women consistently have lower values than the men, leading us to this distribution? And in the last figure, we have a situation of unequal n. So the data points for the second group are clustered at the top end of the range of the data points for the first group. However, it could just be that we've underestimated the variability in that second group because the sample size is so much smaller. And so having more observations in that second group would be really helpful to confirm that we have an accurate picture of what's going on. And I think it's important to emphasize that we can't distinguish between these different scenarios based on the p-values alone. There really is no substitute for seeing the data. So the next question that I usually get asked is, okay, but let's say I know my data are normally distributed. Can I use a bar graph then? And the answer to that question is, unfortunately, still no. There's a couple reasons for this. The first is that the bar graph doesn't allow you to critically evaluate continuous data. And the second is that it distorts our perception of the range of observed values, and it focuses it on whether or not the means look different instead of how much overlap there is between the groups. So if we look at the bar graph, we can see that it's distorting our perception of the range of values in 2 different ways. In some cases, the values close to 0 are biologically meaningful, but in many cases, they're not. The range close to 0 may include values that are never measured or never occur in the data set that we're measuring or aren't biologically or physiologically possible. So in that case, the bottom portion of our graph is really just highlighting what we call the zone of irrelevance. And the second problem is the zone of invisibility. So if we look at the graph, we typically end the y-axis just above the error bar for the highest group. And when we do that, we actually cut off values that were observed in our data set, but don't make it onto the graph. And we call this the zone of invisibility. So when we're thinking about how to show our data, it's important to remember that our interpretation depends on what we see. When we're looking at a bar graph, the reader is a passive observer. I can think about whether the bars look different, but there's really no other information for me to assess. When I see a dot plot or a figure that shows the data distribution more clearly, the reader becomes an active participant. So for example, I can see that there's an outlier in the third group, that the sample sizes are small in these studies all across, or in these groups across the board. I could have a bimodal distribution in the first group, and so on and so forth. All of those details are immediately obvious to me, which helps me to gain more insight into the data. So when we're thinking about data presentation, all data presentation methods are a reflection of reality. Looking at your data in a bar or line graph is a bit like looking at a reflection in a wavy pond. So let's say I developed my hypothesis, I was looking for a duck, and I look at my bar graph and I think I see a duck. But the problem is that the data may be distorted, they're not clear, I can't fully evaluate them. And if I were to have a clearer picture, I might reach a different conclusion. So no one wants to waste time and resources pursuing something that they think is a duck when that thing turns out to actually be an oddly shaped potato. And that's why it's really important for us to select methods that minimize distortion. So at this point, I'm going to turn things over to Stacey, who's going to tell us more about why this happens in some of our work on statistics education.
Thanks, Tracy. You did a great job of illustrating to us what the problem is with some of the current practices in data visualization. And now we want to think about why this might even occur and what statistics education, the role that it might play in creating these types of problems. All right, so this was a problem that we examined recently in our PLOS biology paper, Reinventing Biostatistics Education for Basic Scientists. This paper has been viewed, maybe not quite as much as our bar graph paper, but still almost 20,000 times. And the crux of the paper was really to examine the statistical or the proportion or frequency of using statistical analyses in basic science publications as compared to how often those researchers are actually being trained in statistics. And so what we found is that statistics are really essential for publishing in the scientific literature for these fields, but unfortunately the training is not always required to receive a PhD. So what did we find specifically? So we re-examined our set of about 700 papers in top physiology journals, the same data set that we used in our bar graphs paper to examine data visualization. And now we went back and looked at that data set to investigate how often were statistics actually used in these papers. And we found that by and large, almost all of those papers included a statistical analysis, over 97%. But then we wanted to directly compare that to the statistic education that those investigators are receiving. And to do this, we looked at the statistical education requirements for PhD programs in 80 top NIH-funded physiology departments and found a big gap as compared to how often the statistical analyses were being used. Unfortunately, only 64% of those departments were actually requiring statistics for the PhD. So that leaves this large gap of students who are coming out of these programs with possibly no statistical training at all, even though their field really requires them to use statistics in order to publish in the field. And then even for that 64% that are receiving training, we found that it's often falling short of what they really need because courses aren't really targeted towards basic science, basic scientists. And the reason for that is that usually these statistics courses are not actually taught in the basic science departments. They're really contracted out, you might say, and are taught in epidemiology or biostatistics departments, similar to the department that I'm in. And they're taught by faculty similar to me that generally don't work with very, very small sample sizes, but instead work with what we might consider medium or large sample sizes. And so this causes a disconnect between the instructors who are usually working with larger N, and then the basic scientists who are often working with very small N, not realizing that the problems that they face might be very different. So the instructors teach the statistical concepts focusing on large N that they're used to, not necessarily realizing that they have basic science students in the audience. And so often small sample size issues are not even mentioned because the instructors might not even know that it's relevant. And so the basic science students who conduct research focused on small N take what they've learned from their instructors and apply it in their settings, or maybe modify it in some way that they feel is appropriate for their scenario. But that leads to possible, major disconnects in analysis techniques and creates big problems when it comes to what we see in the scientific literature. And so when we first embarked on our collaboration, and I really learned a lot of details about these problems that we were seeing in the basic science literature, I was actually quite surprised and had told Tracey, this on the bottom of the slide here is probably a direct quote from me, in fact, that if I had known this information before, I would have designed my courses differently. And I'm not alone in thinking this as a statistics educator. And so what are we going about doing to solve that problem? Well, we're designing a course differently, targeted to basic scientists. So when thinking about teaching basic scientists, it's important to really know your audience. And our first area of focus is really the data visualization and the data presentation. Our hope is that if we're able to improve data presentation practices within this community, that the statistics themselves will come second. Once investigators are able to really see and understand the data that they're generating, they'll be motivated to learn the appropriate statistical techniques. We also realized when developing this new curriculum that basic science students have not been completely isolated. You know, they're not doing their research in a vacuum. They have learned, whether it's intentionally through an introductory statistics course that was maybe not designed for them, or indirectly by reading the scientific literature, they have learned statistical concepts, and they may have misconceptions about these concepts. And they may also have missed skills that weren't taught in their introductory statistics course, but are really critical for them in conducting their research. So it's important to really target these misconceptions and missed skills in these types of courses. And our strategy, in addition to that, is also focus on a visual approach to learning to allow students to really see and understand the problems, and also to learn by doing. And to learn by doing, we've created a series of interactive simulation tools that I would like to take a moment to show you a few right now. And the first one, this is a modification from one of our online simulator tools, and is designed to target the misconception that summary statistics are always appropriate, no matter what your sample size. And what we're showing in this figure is 100 random samples, either of size 5, which is here shown in red, or size 20, here shown in blue, that are drawn from a population with mean, denoted here by the black line, mean of 3 in the true population, and standard deviation of what's shown in the gray region, shaded region of the true population. So we've drawn 100 samples from this population of size 5 or of size 20, and then are plotting the points indicate the sample means for those samples, and then the vertical lines indicate plus or minus 1 standard deviation for that particular sample. So you're able to see for the different samples how the sample statistics themselves compare to the true population values. And what we see is that when sample sizes are small, the means and standard deviations can be wildly different from those true population values. So in particular, when n is 5, for some of the extreme samples, we see over here a sample mean that's more than 1 standard deviation above the true population mean. And here's another instance where we have a relatively extreme sample where the standard deviation is less than half of the true population standard deviation. So summary statistics in this setting are not going to be particularly meaningful, because really we don't have enough data to summarize. And so another visual tool that we've created with our online simulator here allows you to investigate how sample size plays a role in our ability to assess the shape of our data distribution. So in this figure, we have populations of 3 different types, either normal distribution, skewed, or bimodal distribution. And we're drawing a sample from each of these populations of size n, denoted here at the top, 10, 20, and 5. And when the sample size is 100, or excuse me, 120 and 5, when sample size is 100, we see that the shape of the data in our sample parallels the true population quite closely. And it's easy for us to distinguish normal versus skewed versus bimodal distributions. As the sample size becomes smaller, once we have 20, it becomes a bit harder to discern whether the distribution's normal, skewed, or bimodal. And when n is 5, all bets are off, right? It's almost, if not completely impossible for us to determine the shape of distribution when we only have 5 data points. And what does this mean for when we want to do something like conduct a normality test on our data? So in this scenario, what if we draw 100 samples? How many of them from the 3 different populations here would fail a normality test? So if our true population distribution was skewed or bimodal, we would expect that virtually all of the samples would fail the normality test, because the distribution is in fact not normal. If the distribution was truly normal, we would expect about 5% of the samples to fail, because that's the type 1 error rate. And when our sample size is 100, that's exactly what we see. However, when the sample size drops to 5, unfortunately, the situation is much more dismal. And when our true distribution is skewed or bimodal, we're almost never actually failing a normality test. The rates of failure are very similar to our type 1 error rate in this situation. So normality tests really aren't working very well for small sample sizes. And so as I already mentioned a little bit before, a lot of these visualizations are adapted from these online simulator tools that we've developed in our suite of simulators called Size Matters that's available at the link posted here, rtools.mayo.edu. So if you're interested in these types of visualizations and would like to learn more, I suggest you check out the link. The visuals I presented here are from Simulator 1. From that series. We have more simulators that are up and coming and should be probably available in the next week or so within that link. But that'll give you a chance to really, test some of these concepts on your own. And additionally, we are in the process of developing an online course for which the simulator is a large portion, and we're expecting that course to roll out sometime this summer. It's called Sample Size Matters, Misconceptions and Graphs and Statistical Analyses in Laboratory and Clinical Research. And right now we're in the pilot testing phase of this course, so please shoot us an email if you might be interested in pilot testing some of these materials. And at this point of the presentation, I'm going to turn it back over to Tracey to tell you about solutions to some of our data visualization problems.
Okay. So we understand more about why there's a problem. How do we go about fixing it? I'm going to go through some of this material kind of quickly. There was a Twitter thread shared in the announcement for the for this particular webinar. And it's also the first Twitter thread shared in the link for the Twitter thread that was posted at the beginning of this webinar too. So this has much more detail than I have time to go over in the talk today, but you can certainly refer to that as you're making graphics to help you decide how to best create your graphics, how to make them most informative and effective, and which type of graphic you need depending on the characteristics of your data. But I'll just quickly review some general principles. So if you're working with continuous data and you want to compare groups and you have a very small or a small sample size, then your best option is going to be a dot plot. And the reason, as Stacey illustrated using the visuals from the simulator, is that summary statistics are only meaningful if you have enough data to summarize. So when we start adding in error bars, or when we use something like a box plot or violin plot that shows more summary statistics that requires us to know about the data distribution, and our N is 5, we just don't have the level of detail needed to get good results with those figures. So a dot plot is the best way to go. And a dot plot is appropriate if their sample size is too small to determine the distribution, or if you have any type of distribution. It really doesn't matter. Your next option is going to be to combine a dot plot with a box plot or violin plot. And this works really well for medium sample sizes. We get some information about the dots, so we can see roughly how many data points there are and assess the distribution, but we also get the more detailed summary statistics that a box plot or a violin plot can provide. And if you're doing this combined graphic, you can also do that with any data distribution. Our third option is a box plot. So at some point, your sample size is going to get large enough that showing all your dots is just going to become confusing, and it's going to create chaos as opposed to providing information. When you reach that point, your sample size is now large enough to move to just showing a box plot without the dots. So you're going to use it for large sample sizes, and there's an important caveat here. If your data distribution is bimodal, you definitely should not use a box plot. And the reason is that those two peaks will be concealed by the box. So typically you're going to end up with your two peaks, at the bottom and at the top of the box plot, and your a little gap somewhere in the middle where you may have less data or no data, and you won't be able to see it on the box plot. So not a good choice. However, there is an option that does work well for bimodal data or any other distribution with large sample size, and that is a violin plot, which looks something like the figure shown here. So that's also a good option that's more flexible in terms of your distribution. And then your last option is a bar graph. So we hope that we've convinced you to move the bar graph to your dead to me list if you're working to continuous data, but that doesn't mean we should ban all bar graphs from existence. Bar graphs are really designed to show counts and proportions. So if you're working with count and proportion data, then we would encourage you to still go ahead and use a bar graph. Okay, so one of the other common questions I get is, okay, so dot plots would be appropriate for my data set, but they can get really confusing, especially when I have a lot of groups in my figures, or I have a lot of different data points and there's too many things going on. So what do you do about this? Well, the good news is that there are strategies that you can use to make a clear dot plot that both allows readers to see the general pattern of response and also gives access to the information needed to critically evaluate the data, which is the individual data points. So the first thing that you want to do is use a jittering technique to make all of your points visible. And here we have a series of strategies for making all of those points visible from least effective to most effective. Best option is to use a symmetric jittering pattern so that your data points are essentially presented in a way that makes it easy for your eye to see what the distribution might look like. The second thing that you're going to do is use emphasis and de-emphasis. So you want to emphasize your summary statistics and de-emphasize the data points. And the advantage of doing this is that it makes it easy for your reader to immediately look at the figure and get a sense of the overall message of your data, what on average is going on. But then you still have all the information that the reader needs to take a second look to see, okay, does that make sense given the particular data points that you've shown? And they can then go ahead and critically evaluate that data on their second look. So how do you do this? Well, the first panel shows an ineffective graphic. In the second panel, we have increased the width to give us more space between the groups and make it easier to see that they are in fact separate groups. And then in the third panel, we've emphasized the summary statistics, the mean or median line, by showing it in black in the foreground, and we have de-emphasized the data points by putting them in gray in the background. Also important to think about, okay, how do we use emphasis and de-emphasis? How do we make effective graphics when we're combining a dot plot with a box plot or violin plot? Well, if you have a small sample size, just showing the box plot is not very effective. We really need to see the data points to get a better sense of what's going on. One thing that some investigators will do is they'll just add what's called a strip plot or an unjittered dot plot, as we see in B. This is sort of helpful. We can kind of tell how many data points there are in the different groups, but there's a lot of overlap and it's still confusing. So our better options are what we see in the lower panels in C and D. And in panel C, I've emphasized the data points by showing them in the front in black. And in panel D, I've emphasized the box plot by showing it in the foreground in black. And I've also done some other things in C and D to make things a little bit clearer. So I've added sample sizes to the axis labels so that I can see how many observations are in each group without counting. I've made sure that my data points are symmetrically jittered and that the width of the box is proportional to the sample size to give a little bit of information. And then I have one group that has a really small sample size, only 4 data points, and it's not appropriate to show a box plot for that particular for that particular group. So I've removed the box plot from the graph for that group. The next question I get asked is, does it matter how you do your analysis? And the answer to this question is yes. Ideally, we want the figure structure to match our analysis. So let's say we have an experiment where our goal is to compare 2 different kinds of mice. And when we read the statistical analyses, we see that t-tests were used to compare values for our dependent variable. And we had 3 different dependent variables, marker A, B, and C. What a lot of investigators will do is that they will combine those 3 biomarkers into the same figure. And this is confusing. It sends mixed messages because as a reader, I look at this and I think, well, they must have intended to compare the biomarkers as well as the wild type and knockout mice. And now their stats doesn't really make sense for what the figure shows that they're doing. So if we had done better design of this figure, we would ensure that the structure aligns with our goals. So we would have used separate panels for each of our different biomarkers, and that would allow us to see that the goal is really to compare our 2 different kinds of mice and that we weren't interested in comparing the biomarkers to each other. Statistical reporting is also a problem that we don't have too much time to go into today, but those of you in the basic biomedical sciences will know that statistics sections often read data were analyzed by t-tests or ANOVA and don't provide any additional information. So we have a new paper out in eLife illustrating why this is a problem and also showing solutions as to how to fix it that has a lot of visualizations to help you understand both of those things. So we would encourage you to check that out as well. The last part, I'm going to talk a little bit about some of our tools for creating interactive data visualizations. So if we think about the way scientific publishing has evolved, basically we've taken Adobe Acrobat or whatever PDF reader you're using, and we've used it to replace the printing press. And this is a bit of an odd choice because if we think about the incoming generation of scientists and even those who are now in the field, we're very used to using mobile devices and lots of apps and having all kinds of information that we can easily manipulate and transform and adapt according to our needs and interests right at our fingertips. And yet that process that we go through in our normal daily lives hasn't really really influence the way that we visualize data in science. So the goal of this area of our research is to think about what kind of tools do we need to transform scientific publications from static reports into interactive data sets. And one of the tools that we've introduced is an interactive dot plot tool. So this is freely available online. I will give a very quick summary of it, but if you go to the website, there is a link for video instructions. And if you just click on that link, you'll be able to see a video that demonstrates how you use the tool. And it's quite short, I think it's about 5 minutes. So there are two options for entering your data. You can either upload a data set or you can enter your data manually. So if you're entering your data manually, you'll tell it the number of groups, the number of variables, you might list a size for each group and names for your groups as well as your variables. There are instructions that tell you how to format your data set. If you don't feel like doing that, you can just click on this button that says download template CSV file, and it will download a file with all of the information that you've entered, with everything structured the way that the app wants the information to be structured. So you just have to plug in your data and then you can go ahead and upload. The app allows you to create all of the different types of graphs that we have outlined here. So you can do dot plots, box plots, violin plots, and combinations of those. And switching between graphs is very easy. You just click a button for the type of graph that you want. And then depending on what button you click, you'll see some different options here with different ways that you can formulate that graph. So adding dot plot to your box plot, so on and so forth. In addition, because our app is also designed for educational purposes, it will even allow you to show bar graphs. However, if you show bar graphs, you will get this nice pink warning here that pops up saying that the bar graphs are intended as a teaching tool and aren't intended for scientific publications. And we have very kindly provided you with some references illustrating why you shouldn't use bar graphs for publications with continuous data. The other feature of the tool is it allows you to include a subgrouping variable. So let's say I wanted to, I pooled my males and females in my data set for analysis, but I want my users to be able to see the data from males and females separately. I can include that as a subgrouping variable, and then readers can either view everything together, or they can have the option of color coding the two different subgroups. They can show them side by side or all together. And I could even, if I just wanted to look at males or females, I could remove one group from the graph by unchecking the boxes here. The additional thing that we offer is the opportunity to show clusters of non-independent data. So this might be things like replicates or mice from the same litter. And the clustering function will work for up to 6 clusters. It will color code the different clusters so you can easily tell which data points go together and are non-independent and grouped. And it will work whether your clusters are arranged between groups, within groups, or between and within groups. There are some additional features for publications, so you can save any static graph that you want to include in your paper, and you just enter the name, so say the name of the figure in your paper, and that figure will now appear in the build menu. So when readers come into the graph, they can either explore the entire data set or they can click on a particular graph in order to look at that in more detail. And you can also download a few things, the CSV file if you just want to see the data, or you can download TIFF files, and this allows you to match your static figures in your publication with the interactive graphic. And then the last thing that we allow you to download is an XML file containing the interactive graphic. So there is no data stored on the site ever. Anything that you enter is gone as soon as you leave. But if you put that XML file in the supplement of your paper, your readers can then take that XML file, re-upload it into the site, and view your graph as you created it. We also have created an interactive line graph, and I'm not going to spend too much time on this today other than to say there's also a video on the website that you can view if you're interested in finding out more about how to use this tool. And the tool allows you to do 4 things. The first is to examine different summary statistics. The second is to display lines for some or all individuals in each group, so we can tell if everyone is responding the same way, if there may be subgroups of, say, responders and non-responders. We can also look at a subset of groups, conditions, or time points, depending on what's interesting to us, and we can view change scores for any 2 conditions or time points. So where do we go with all of this? Well, right now, it's critically important for us to be changing data presentation for continuous variables in small sample size studies in order to promote transparency and allow us to critically evaluate the data. So the sumo wrestler we see right here right now is the use of bar graphs for continuous data, and this little guy over here is the use of more informative graphics. We need to flip this around so that this is the more informative graphics and bar graphs of continuous data are being used rarely, if ever. What are the things that you can do to help with that? Well, the first and most obvious thing is to banish bar graphs from your papers, talks, posters, and presentations. For those of you who are reviewing or acting as editors, request figures that show the data distributions, give people citations and resources to help them make better graphics and understand why this is important. You can also talk to editors in your field about improving the quality of data presentation in their journals. And finally, you can organize data presentation and statistics training for trainees, junior investigators, and senior researchers. And you may want to collaborate with your statistics instructors or other people in your department in order to do this. And lastly, I think it's important to emphasize that this work really shows how important it is for all of us to be a part of solutions when we're thinking about improving rigor and reproducibility in our own fields. So this whole bar graphs project and the meta research program that I now have came out of me wanting to solve a problem in my field that I felt was holding the field back. And many of you also probably have similar challenges or limitations in your own fields that you're concerned about. So what can you do? The first thing is to read meta research studies to learn about existing problems, discover better practices, identify the problems in your field, and see how other fields may have solved similar problems and whether you can simply import their solutions to improve practices in your own field. And then the second thing for those of you who are more interested is to build a team and perform your own meta research studies to help solve problems specific to you. And with that, I think that's all we have for today. So we'll open the floor up to questions. We do want to thank a couple of people. So Ethan has been extremely helpful in creating the online simulators that Stacey talked about. Oscar is one of our trainees here who's been very helpful with conducting some of our meta research studies, and Vesna Garovic is my mentor for the BIRCWH program. And then our collaborative team at the University of Belgrade Medical Center is also critically important. So Natasha Milic is the chair of biostatistics there, who is involved with all of our meta research and our statistics education. Marco is the programmer who created the interactive tools that we hope that you'll check out and experiment with and send us feedback on. And then Jelena is also at the University of Belgrade and has helped with our meta research studies. So at this point, I think we can open things up to questions.
Thank you so much. That was an excellent talk, and you gave a lot of wonderful resources. I think one of the coolest parts about what you presented is that a lot of this is the outcome of multiple disciplines working together to solve a problem that was identified. So I can't imagine either of you could have envisioned talking on this topic, years ago. But it's wonderful, and I think the resources you provide are fabulous and worth checking out. A common question is, in addition to what you showed us, which was a lot in terms of links and potential, an app to try out, any other favorite resources or software that either of you like to use regularly when you're working with your data?
Do you wanna?
So, well, we do have in the resources, I think Tracey has tweeted out, we have a lot of that available. I, in my own research, use R for making or conducting statistical analyses and conducting or creating graphics. We do have our code to create dot plots and other alternatives to the bar graph for those who would like to use that as a tool. But we have other tools as well. Besides the interactive graphics that Tracey presented, we also have code for some other statistical packages too, and even Excel templates. I don't know if I should even, as a statistician, are they going to revoke my credentials if I even tell you that I helped create an Excel template? Yeah, so I suggest you check out the research page. It looks like Kayla has sent out Tracey's Twitter feed with the resources.
Yeah, so in the main tweet that was sent out in the announcement for the talk, the fourth tweet in that thread, I believe, has a huge list of all kinds of different data visualization resources that you can use. I don't think it's essential to be particularly hung up on finding any given resource. I think what's most important is you find one that works well for you and for the types of data that you work with routinely. So whatever, whether it's an online tool or whether it's a program that uses code or the stats software you're used to using, whatever it is, that's fine. The most important thing is the graph that you get out of it is informative for you and for your readers. And as a reviewer, I could care less how you created it.
It's very helpful. Do you have any thoughts or tips on presenting to the non-scientific community? So what I mean by that are like patients or policymakers, perhaps the media. Any thoughts on those, the lay people who maybe are not used to doing biomedical sciences, or any thoughts on that and tips and tricks?
So I think that's something that we do a lot less of. We do a lot with communication strategies, but we really focus on communication strategies for scientists within the scientific community. Some things we've learned from that: it's really important to understand who your audience is, and not just a superficial overview of like, we target biomedical scientists who work with small sample size data sets. That's not really meaningful. We actually, when we work with our collaborators, we have a document that is now more than a page long. It is in single space font, or single spaced 10-point font, bullet points, that outlines everything we know about our audience that we've learned from reading hundreds of papers and meta research studies. So we know, what types of software they're using, what types of data set are they working with, what sample sizes, what study designs, what mistakes are they used to seeing as part of standard practice. We know all of those types of things, and we don't start writing a paper until everyone is on board and understands everything that's in that document. And I think developing, whether your audience is scientists or whether it's people in the public, having that kind of intimate knowledge of how people think about things is really essential to putting together your content in a way that's going to resonate with them. The other thing that
and not in a way that they might misconstrue.
Yes.
Or misconceive. Because I think a lot of the problems that we outlined here might be specific to, small sample size studies or basic biomedical sciences, but there are similar problems, right, that exist in communicating statistics and visualization and graphics to the lay public, that the same principles still apply, that it's very important to show your data in the most truthful way possible, and not conceal certain aspects just because you think that your audience might not understand it.
The other tip that I think is really important, if you can have someone who is part of that audience that you're targeting give you feedback on your materials and tell you, this is what I thought, or this is how I reacted, or this is what didn't make sense, that will help you really strengthen what you're working on. And then the third thing I would say, we use a lot of visuals. So we actually design every paper around our visualizations. We agree on our most difficult concepts. We design a visual for everyone, and then the papers are basically words and data to make all those visuals fit together and make sense. That's a very handy approach when your audience doesn't know nearly as much about a topic as you do. And so we use it a lot with basic biomedical scientists who we know have limited statistical training, because we could write a bunch of words and formulas, but it's much more effective just to put up a figure. And we will test our figures out on people and say, in 10 seconds, what can you tell me based on this? Mm-hmm. And they will either tell us what we want, what we were hoping they would tell us, or they tell us something completely different. And if they tell us something completely different, we have work to do. Mm-hmm. So I will actually send out in my Twitter feed later a blog post by a colleague of mine, Helena Jambor in Germany, who has written just a really nice blog post on how to design effective visualizations that I think would be beneficial for people who are interested in scientific communication, either for scientists or for the general public.
Oh, that's excellent. So this webinar is being recorded as a resource, and you mentioned, this upcoming online course maybe this summer. Any other YouTube videos or podcasts that you'd recommend to our audience that could help further inform them?
So I provided some links in the thread that was posted at the beginning of the webinar to a Science of Science reading list. So that's a really good meta research resource that covers a lot of different topics. The other thing I would raise as a possibility for those of you who are early career investigators who are really interested in changing the way science works, the eLife Ambassadors program has a call open right now for applications. And this is a one-year program. It's volunteer. You do it, in addition to your other stuff. And it basically brings together young investigators who are interested in any aspect of how science works. So I run a meta-research team through that, and that meta-research team created the Science of Science reading list that I just mentioned. We're also working to do a meta-research project on the quality of reporting for images that will be peer-reviewed and published in a scientific journal. There are other groups that are organizing reproducibility workshops, that are organizing, preprint journal clubs, and there's all kinds of different initiatives. So I think if you're interested in getting involved with something a little bit more substantial and rethinking the way science is done, or simply just learning a lot from people who are very active in this area, that's something that I would encourage you to think about applying for.
Awesome. Great. Anything else you'd like to share, either when it comes to metadata or just how you got to this field, or any other things that you'd like to share with us BIRCWH scholars?
I would like to highlight how our collaboration started, I guess, because it did start within the BIRCWH program. And as a piece of advice, to make sure you keep your options open and know when, be open-minded to the possibility of a good opportunity, because this was something that I had never even, you mentioned when we started the talk that, probably 5 years ago, we wouldn't have imagined that we'd be sitting here today talking about this particular problem in our set of solutions. And that is so very true for me, that I never envisioned that this is where something could have went. I never even really, I shouldn't say never, but I didn't fully realize that the extent of this problem and the role that I played as a statistics educator in the problem and the role I could play in the solution, and how important that when Tracey first approached me about this problem, I literally told her, "You can't be serious," or something along those lines. Like, people can't be doing that.
Yeah, it took me 5 to 10 minutes to explain to Stacey how you would make a bar graph of continuous data, because she just didn't understand what I was talking about. And I think the response from my other statistical collaborator was similar. I went to her with the data that I had collected on the prevalence of bar graphs in physiology journals, and she said, "I don't believe you." And I said, "I have data. You cannot believe me." And she said, "I understand, and if what you're saying is correct, that's terrible, and we should really do something about it. But I just can't believe that an entire field of people would be making, this critical an error." And my response to that was, well, if you're following standard practice for your field, you're never going to be asked to justify it. Mm-hmm. So in terms of advice for people, I would say, I think one of the things that this project has illustrated to me is that it's really possible to have an impact on changing the way science is done, which will help your normal research area, but also help all of science as a whole. So for me, it's really expanded the horizons of the impact. When I think about the impact of my work, it's expanded the horizons there. And I will also say that I started this project after being frustrated about bar graphs for 10 years. And if I had known that I could have addressed the problem and started fixing it 10 years sooner, I probably would have done that. So I think don't assume that just because something is common in your field, if you know it's a problem, then figure out how you can work with your colleagues to go towards solving it. And if you're interested in doing meta research, definitely consult with people who've done it, who can give you good advice on how to design your studies and how to take the issue forward and how to create interventions that might actually work, and then test those interventions.
Great. Now, you both have an interest in sex-based science and studying that, and you showed with the app an example of stratifying with male and female. Do you have specific visualization methods that you prefer to use when demonstrating sex differences in data sets?
So do you want to? Sure. That will depend a little bit on the differences you're trying to show and, the structure of your study design and the problem. But I think one of the visualizations we highlighted, the potential for a bimodal distribution in particular, can pop up often when you're looking at sex differences. where you definitely at least want to make sure that the sex is present, right, in your graph, whether it's as a separate subgroup, so maybe separate panels, or you know, separate groups in your graphic, or whether or not it's colors, you know, like red for women, blue for men, or different data symbols or something like that. You want to make sure it's emphasized. But I also think then if it's a situation where you might be expecting something like a bimodal distribution, you want to consider a type of graphics that will allow you to show that. So maybe like the violin plot or the dot plot as well, if you have a smaller set of sample sizes. But I'd think carefully about how your study is designed, and whether or not it's more important for you to emphasize the sex differences as you know, separate groups, or whether or not you want to group them together and illustrate the sex difference by like color or symbol. And I don't know if that's similar to what you were going to say, but.
Well, I was waiting for you to answer because I studied the pregnancy complications of preeclampsia,
Yeah.
and there are no pregnant men in my studies, unfortunately.
Sure.
But if you find me a pregnant man, I will study him happily.
But yeah, I think, like, you know, all the principles we've discussed apply. You know, at the end of the day, you're still trying to illustrate a difference, right? Whether it's a sex difference or a difference in another type of group. So the really important part is that it's very clear on your visualization. So things like color, emphasize that. And
yeah, and the other thing I would say, it's not just about showing differences. Sometimes it's equally important, in fact, all of the time, it's equally important to show they're not different. You know, if you made the decision to pool males and females in your data set because they weren't different, why. You know, if they can see that data set and look at it and say, yeah, okay, there's no difference here, it saves, it answers a lot of questions for the reader, and it may save you some rebuttal. The other thing that I would say is, if you are using color, there are a lot of free tools that allow you to see what your color palette will look like for a colorblind individual. So there's a free tool called Color Oracle that you can download in like 30 seconds. Once you have it downloaded, you click a button and it will change your entire screen to what a person with colorblindness would see. So anytime you're using colors in your visualization, whether it's a graph or a data figure or a ultrasound image or a microscopy image, using that kind of a tool is really helpful, just to make sure that your information is accessible to everyone. Because around 8% of men of North American or European ancestry are colorblind, and 0.5% of women. So it's not a small portion of your audience that will be missing things if your color palette isn't colorblind safe.
Do you suggest in that case, maybe even for dot plots, trying different shapes? Do you ever, I'm just curious.
Well, I have a little bit conflicted feelings about different shapes. A lot of times I think it's just unnecessary distraction. If your groups are appropriately labeled, then there isn't really a reason why you need to have a different shape for every group in your data set. So it really depends. If color is an option for you, I think that's a lot more accessible for people, as long as you have a good color palette, than differences in shape, which can be a little bit harder to detect. So yes, shape is a way of distinguishing between different things, but I think it's really important to think about what is the overall message of my figure, and then based on that, you know, is my figure really getting that message across, and removing anything that's extraneous or that's not really adding additional information. So if you think about all the dot plots that I showed in the talk, I was using the same shape across the board. You can still clearly see what's going on based on the axis label. So having a different shape in those particular circumstances wouldn't have really added much value.
Right.
I also think it depends how many types.
Yes.
Because if you're only talking like 2 groups that you might want to indicate on a shape, that might be less visual clutter, you might say, versus like some of the clustered graphics that Tracey showed, where there were, you know, maybe 4 different colors we were using to illustrate. That would be a lot messier and harder to digest with different symbols.
Yeah, fair enough. Well, thank you to both of you for taking the time to present these very useful tools and advice. I'd like to say to all of our listeners, stay tuned for an announcement on the next speaker and date for our next webinar in the BIRCWH program. And please complete the survey after this webinar. And again, thank you both for sharing your knowledge with us all today, and all the wonderful resources you've shared with us I'm looking forward to exploring and using them myself. So thanks again.
Thank you.
Thank you.